Hi, my name is Christine Byrne, and I've chosen to do prompt 1 for exam video 6, explaining positive inducible transcriptional regulation in prokaryotes. Before discussing the specific mode of regulation, let's understand an overview of how prokaryotic transcription works. To begin, we have a segment of DNA composed of genes and clusters of control mechanisms that make up what's known as an operon. The ultimate purpose here is to allow an enzyme known as RNA polymerase bind to the operon at the promoter region and move along the DNA sequence, reading and transcribing the codes of structural genes that will later create gene products such as proteins. There is one more key element of the operon known as the operator, which is a crucial factor in regulating whether or not transcription will occur. The operator is the area where regulatory proteins bind, and depending on the type of protein that binds, transcription will either be stimulated or inhibited. A regulatory protein that stimulates transcription is known as an activator, and this is known as positive control. In the case of positive control, activators actually bind to operator regions upstream of the promoter known as activator binding sites. On the other hand, a regulatory protein that inhibits transcription is known as a repressor, and this is called negative control. To break it down even further, there's also a small molecule that controls the functionality of the regulatory protein itself, known as an effector. The way this works is that when the effector attaches to the regulatory protein, it alters the shape of the protein, allowing it to either attach to the operon or inhibit it from attaching to the operon. In the case of what's known as inducible control, the effector, in pink, changes the shape of the regulatory protein, in yellow, such that it can attach to the operon, turning transcription on. In the case of what's known as repressible control, the regulatory protein is normally attached to the operon and transcription is normally on, but the effector changes the shape of the regulatory protein such that it causes the protein to no longer attach to the operon, thus turning transcription off. Now that we know about positive versus negative and inducible versus repressible control, let's talk specifically about positive inducible. In this mode of transcription, the regulatory protein is an activator and transcription is activated when the protein is bound to the DNA. In order for the protein to bind, the small molecule present must change the shape of the protein to allow it to attach to the operon. This bound protein then allows RNA polymerase to recognize its promoter region downstream and initiate transcription. Here's an analogy to better explain these moving parts. The purpose of this lamp is to provide light, but it relies on two major factors that allow that to happen. The switch must be flipped up to the on position, and this plug must be connected to an outlet. Let's say that when plugged in, the switch is then flipped to the on position. If the switch represents a regulatory protein, the plug represents a small molecule, and the light represents transcription, then this is an example of positive inducible control.